Um, as a eukaryotic organism, we have to be able to churn on, right, and churn off our genes. Inside of our brains, we have brain cells, and inside of our liver, we have liver cells. And the liver cells should only be producing and activating liver genes, and the brain cells should only be producing brain genes. Think of all the cells in your body like all the restaurants in Los Angeles. There are different types of cells, and there are different types of restaurants. But inside of every restaurant, or inside of every cell, is all of the DNA that contains all the genes for the entire human genome. So imagine walking into a Thai restaurant, and in the back, in the nucleus of the Thai restaurant, is all the recipes for all the food of every single restaurant in Los Angeles. But they only churn on the Thai recipes because it's a Thai restaurant. So my brain cells only produce brain proteins. I'm not going to produce insulin in my skin. I'm going to produce it in my pancreas. I'm not going to produce dopamine in my muscles, right? I'm going to produce it in my neurons. Although I have neurons in my muscles that run through my muscles. So there's this gene expression that we have to be able to churn on certain genes and churn off. And it's different in bacteria as it is with us. And it's because we have specialized cells that makes it extremely difficult to be able to regulate all that. So we have to have highly complicated gene expression and regulation. So let's talk about mutations. Some mutations can affect a single gene, and that's what your assignment is going to be today. And some can infect the entire chromosome. And the ones that affect the entire chromosomes have huge effects on the phenotype of the organism. So a mutation is a change in an organism's DNA, and sometimes that change can be for the good. Now, mutations that affect a single gene, so it happens on one gene, usually happen when we're copying that DNA. And so we can have different types of mutations. We can have point mutations that occur at one point, right? And some examples of those we're going to get into. Now, if we have mutations that occur on the entire chromosome, which is what we actually got tested on last class, that's going to have some huge implications. And that usually happens during meiosis, when those chromosomes have to line up next to their homologous parent. So let's talk about the gene mutation that I'm going to test you on. And this is sickle cell anemia. And so as you see here, this right here, this is a sickle cell. And the reason why it's a sickle cell is inside of all of it, this is kind of the normal shape of a red blood cell right here. Inside of these blood cells are a bunch of hemoglobin proteins. And there's like 250 million of them. And hemoglobin, let's say here's a hemoglobin protein, here's a hemoglobin protein. This is what transports oxygen, these proteins. And they're usually kind of spread out throughout the cell. But in sickle cell, the hemoglobin proteins have a mutation. This is the first 1, 2, 3, 4, 5, 6, 7 amino acids in a 146 amino acid long polypeptide that is part of hemoglobin. Right there at one point, that's why it's called a point mutation, the A is substituted, this is called a substitution, to a T. And if you translate GAG into messenger RNA, right, GAG is CUC, that codes for glutamic acid. But if you mutate this is the DNA right here. If you mutate the DNA from GTG, that's going to code for CAC, and that brings over valine. And so now the hemoglobin is shaped differently, and they tend to stick together in these long rods. So the hemoglobin kind of clumps together. And these long rods of hemoglobin build up in the cell and give it this sickle shape. And so sickle cell anemia is an example of a point mutation, specifically a substitution, where at one point we substitute an A for a T when we're copying our DNA. Yeah. And people can inherit those. They become permanent once you inherit them. Now, frameshift mutations happen at one point, but it involves either inserting a letter or deleting a letter. And so remember that in this case right here, see how there's two G's right here, guys? Now look at the frame. It's GAT, which codes for leucine, CTC, which codes for glutamine, and then AGG, which codes for serine, and then CTA, which codes for aspartic acid. Or is that asparagine? I think that's asparagine. Not asparticus. Now look what happens when we delete one of those G's. GAT is not affected. Anything before the deletion is not affected, so still leucine. So we're still good. CTC, still glutamine, still good. We deleted it and shifted it over. AGC, AGG, and AGC actually code for the same amino acid. That's a silent mutation for that amino acid, because this last one is different, but it codes for the same amino acid. But here's where we have a difference, right? CTA, this is now TAA, and this codes for a totally different amino acid. When you have a different amino acid, that's called a missense, when it codes for a different amino acid. It's like this. The fat cat ate the rat. If we were going to delete a letter, let's delete that A. Oops. Then look. Now look at how the reading frame has shifted. The, fatka, ata, tet, her, ah, right? That is totally not the way that sentence should sound. Oops. Let's just do this, right? Let's try that again. Ugh. Oh, this is all jet. There we go. Mitch, do you need to take a step out, or are you good? Now, before I delete it, now I want to add. Let's add another T. The fat tika tat het hera, right? These are called frame shifts, right? They totally affect the order of amino acids after the insertion or deletion. Chromosome mutations, I already tested you on last unit. But there's, no, I'm not going to test you again, but just to remind yourself, these usually happen during meiosis, and there's 4 types, where we have a duplication, where we duplicate a gene. We have translocation, where one part of a gene from like chromosome number 1 goes on chromosome like number 4. And so you have this kind of switching or translocating of genes. And then there's the other 2 that are not even mentioned here, the inversion and deletion. Now, mutations may or may not affect the phenotype, depending on whether or not it changes the shape of the protein. Chromosome mutations have a big effect. I mean, look at this turtle. There was some, like a whole chunk of DNA might have been duplicated or translocated or deleted or inverted.
Looks like that, like photo booth, like effect where
It's one of those. It mirrors, yeah. That's a Snapchat of a turtle. Some gene mutations do change the phenotype. Sometimes you have an early stop codon. This is called nonsense. When you change to like UGA, if there's a mutation and it's supposed to keep on going, this is a stop codon. So if the amino acid sequence is supposed to be like 150 or 200 amino acids long, and on the 4th amino acid it says stop, then that's done. The ribosome detaches, and you have this tiny little chunk of polypeptides. That protein is not going to work. It is just utter nonsense. Anytime you affect the protein's shape, right, especially active sites of enzymes, you're going to affect its function. unless it's a shape or a part of the protein that is not significant or involved in the function of the protein. Any mutation that does not affect the phenotype, so like you change a amino acid and it's the same amino acid, or you change the shape of this part of a protein but not the part that matters, which is the active site. It's like me shaving off the back part of my key, not the grooves that fit into the lock. If you shave off the back part of a key where you grip, it's not going to affect the key from opening up your door. It's only if you affect or change the shape of the key that is those grooves. It's the same with proteins. Now, where do mutations matter? Well, if you have a mutation on any of the body cells, we call those somatic cells, it does not affect your offspring. If you have a mutation on your skin because you've been out in the sun too long and you get skin cancer, you've mutated the DNA of your skin cells, you can't pass that on to your children. But if you have mutations in your sex cells, these are your gametes. If you have mutations in your sperm or eggs, then those mutations are going to affect your offspring. This is a mutation, right? A single mutation that affects the hair growth in the specific follicles that we have. That we have two types of hair, the soft hair that comes off of our skin, and then we have this terminal hair that comes out of our head. And it doesn't take much in terms of genetic mutations to be able to get phenotypes that look like this guy. There's actually multiple individuals that have this specific genetic mutations. To shave your face like that, try shaving your forehead every single day. It's much easier just to let it grow. But it goes to show us something. It goes to show how very little we have to change our DNA for us to see kind of where we evolved. It's not a huge amount of tweaking of those letters to see how much we have in common with other apes. Anyway, we kind of got cut off talking about how you don't have to change much to see how closely we are related to other apes, because as human beings, we are apes, along with gorillas and orangutans. And so you don't have to change too many genes to see the similarities that we have, since over 97% of our DNA between us and chimpanzees is identical. It's the same. This is Windy the Whippet. Windy the Whippet has a double mutation on a gene called the myostatin gene. This gene is in charge of the thickness of our muscles and growing all the proteins that make our muscles. So she has a double mutation. And so sometimes this happens when DNA replicates itself, and so they can cause mutations. That's typically where a lot of our mutations come from. They're just random duplication errors. But things that cause mutations are called mutagens, because they generate, gen means generate, mutations. UV radiation, so you get skin cancer, can kind of, think about it like microwaving the DNA, causing it to mutate. Chemicals from cigarette smoke, your environment, asbestos is known to cause cancer, obviously. Things that are in your food, these also can cause cancer. In fact, some of the chemotherapy, the drugs that they put into your body, some cancer drugs, are actually mutagenic, and they go into cancer cells and mutate the DNA, killing the cancer cell that way. All right, so real quick checking for understanding, then we're done. Mutations are changes in the DNA. They could be bad, they could be good, or they could be silent. They could have no change at all. Some causes of mutation: UV radiation, the food that we eat, our diet, chemicals, or replication errors. The difference between a point mutation that is a substitution, which is what sickle cell anemia is, and a frameshift is that a point mutation or substitution only affects one amino acid. Whereas a frameshift, which is by adding or deleting, or another word for adding would be inserting, or an insertion. This is going to affect every amino acid after that because it shifts the frame of reading. Mutations only impact the organism's phenotype if it changes the shape of the protein, and that protein has a different function or loses its function. The different types of chromosomal mutations, even though this is not going to be on the test, is insertion, deletion, duplication, and translocation. And in order for mutations to affect your offspring, they need to take place in your gametes, which are your sex cells.